I will go ahead and get started then. So good afternoon, everyone. My name is Sherry Shia. I am a bioinformatician in the Mayan lab, and today's lecture is just going to be an introduction to enrichment and pathway analysis. There's going to be three main sections in this lecture, which are going to go over the concept of enrichment analysis in general, as well as two popular methods for computing enrichment analysis, which are the GSEA algorithm and the Fisher's exact test. So I'll go ahead and start with an introduction to enrichment analysis, and I'm going to try to give some more background on what it is and why we use it. Essentially, vast amounts of omics profiling data are published each year, and oftentimes this data is focused on specific physiological or pathophysiological conditions. With that data, one thing that we want to do is obviously identify common functions for genes and proteins, or potentially explain molecular mechanisms underlying a specific phenotype at the cellular or organismal level. And this is something that we can't do just by looking at one specific gene. So we do want to look at, sets or groups of genes. And so for that reason, we have a method that's usually called gene set enrichment analysis, or pathway enrichment analysis is also a term that you'll hear referred to as sometimes. And so generally, we know that omics studies will focus on a specific process, disease, or compound or condition. And then through some type of molecular profiling, you can generate a signature, which I believe you heard about that last lecture. And then from that signature, you can actually turn it into a ranked or an unranked gene set. And here, when I mention molecular profiling, usually it just refers to one of the following. So gene or protein expression profiling studies, such as RNA sequencing or microarray, and these would usually result in a type of gene signature. There are also GWAS studies where you can get a set of genes that are all associated with a specific trait, or ChIP-seq studies where you can find the target genes of transcription factors. And of course, there's many other types of omics assays where you'll get different results that will usually always result in some sort of up or down gene or protein sets. So earlier, I kind of used gene set to describe just any collection of genes, but generally when we say gene set, we refer specifically to an unordered collection of genes with some shared association. So this is different from an ordered signature or gene list. And you can also kind of think of gene sets as like this bag of genes. In this example on the screen, we can include both the most highly and lowly expressed genes from our signature. But we could also create two gene sets from the highly and lowly expressed genes separately, and then those groups of genes would all be just gene sets. We can then combine several different gene sets to create a gene set library. And this is just a collection of multiple related gene sets, such as those from a single resource or generated from a single data set, or gene sets that correspond to very related biological terms. And one well-known source of gene set libraries is the Gene Ontology, or GO. And here I just have an example of several gene sets related to carbohydrate metabolism. And you can see our gene set from the last slide that corresponds to the GO term for glucose metabolism in the blue down in the bottom left corner. And then some of its quote unquote parent gene sets and other related gene sets. And it's important to know that some gene sets may be subsets of other gene sets, and then some gene sets may just overlap, and some gene sets are just completely discrete from each other. And then the last term I want to introduce in the section is the background, which is the context of which genes should actually be considered when you're doing gene set enrichment analysis. Basically, you need to know what the possible genes or groups of genes are that could be significant to your analysis or your study before you can actually identify what is significant. Generally, for the background, you can and should use all genes that were measured in whatever assay you initially used as the background. There are some assays that don't measure all possible genes. One example of this is the L1000 assay, which you may or may not have heard of before. And this assay specifically only gives expression values for about 12,327 human genes, which we know is less than what you could sequence in traditional bulk RNA-seq data. But in this case, you should use only those 12,000 or so genes as a background because you have no way of knowing the expression of the other genes. One common mistake that people tend to make sometimes is thinking that they should reduce the number of genes in their background if they're doing cell type or tissue-specific analyses. So if you consider studying gene expression patterns in brain cancer, you might be tempted to reduce your background set to only look at genes that you know are expressed in brain tissue. However, when you do this, you're kind of running this risk of missing any critical genes that are aberrantly expressed in brain cancer that may not be normally associated with a normal brain tissue expression. So it's important that you include as many genes in your background that you were able to measure and that could potentially have been differentially expressed in your initial analysis. Now that we've defined some of the key terms, we can begin the actual steps for enrichment analysis, which, as we mentioned earlier, starts with analyzing a set of genes or a ranked list of genes obtained from some profiling omics study. So for example, perhaps you're interested in the role of a certain gene, so you obtain your samples for the knockout of that gene, you perform some profiling, and then you can compute a signature for that gene knockout. The next step would be to compare that gene set or ranked list that you just obtained against a gene set library. For your knockout gene study example, you might want to compare your gene set or list against different pathways that you suspect that gene might be involved with. And as I defined earlier, a gene set library is just a set of different functional terms that are each associated with a set of genes based on some prior knowledge or prior studies. The final step would be viewing your results so that you can identify the terms that correspond to gene sets where genes from your input set or ranked list are overrepresented. And you'll notice that I use the term overrepresentation or overrepresented and not overlap, and that's because there is a key difference between the two terms. So overlap looks at the exact number of unique shared genes, pretty straightforward, except that it is a bit biased towards longer gene sets. So you can look at this example I have on the screen, where we're comparing the same input gene list against two different pathways, A and B, and the genes associated with those two different pathways. If you look at just the overlap, the number would seem to suggest that pathway B is, more enriched or more relevant to our input genes. But intuitively, we might think that pathway A is more promising because of its higher specificity, so to speak. And in general, both of those could be true. It could be relevant to both pathways. Neither pathway could be relevant. It's hard to say. But when you're comparing against a lot of different gene sets and you have larger comparison gene sets than others, it's difficult to say whether or not that overlap is truly representative of the actual biology, or if maybe pathway B just has higher overlap with your input genes because there are just more genes in general in pathway B that could potentially overlap. So that is why instead of overlap, we use overrepresentation. And this is also kind of synonymous with enrichment. And this basically looks at whether genes from the comparison set are present more than you would randomly expect in your input gene set. And this does usually require statistical testing. And the test usually, the null hypothesis is that the genes from the comparison gene set are not more differentially expressed in your input data than would be expected by chance. With that being said, there are many, many different tools and algorithms that already exist for performing enrichment analysis. I have put up just a few of some very well-known papers and tools that are already published. Each of these tools will have their own method or computation for enrichment analysis. So in general, there's a lot of different methods for doing this specifically. I will talk more in detail about two methods, gene set enrichment analysis and the Fisher's exact test, which is used in Enricher later on in this presentation. But feel free to read more about any of these other methods, especially if you're already familiar with some of them. But generally, as kind of a wrap up, enrichment analysis results will almost always produce some sort of information that highlights the biological themes underlying the gene set or the ranked gene list that you input into the program or algorithm. For example, which known pathways are evolved, which biological processes are disrupted in your data, what cell or tissue types are represented, and also which gene or protein expression regulators are implicated. So you can ask many different questions with enrichment analysis results based on the type of comparison gene set that you're selecting. So kind of as a final summary of what we just went over, enrichment analysis helps us identify common biological functions for a given list of genes or other molecules obtained from an omics study. The examples I used in this slideshow so far have all been on genes, but technically you can also do enrichment analysis with proteins, metabolites, drugs, or other biological entities, given that you also have comparison sets of those same entities to run the enrichment analysis against. Examining sets or ranked lists of genes or other biological entities can lead to more meaningful and robust results than examining just individual genes. And also many different tools and algorithms already exist for performing this type of analysis. So definitely feel free to explore and read more about it if you're interested.